In this lesson, we want to look more closely at the function of hemoglobin and examine other factors that relate to its oxygen binding affinity. It should not surprise you that pH has to do with the oxygen binding affinity of hemoglobin. This is referred to as the Bohr effect and relates not only to the effects of H+ concentration, but also that of CO2. As we examine the equilibrium reaction shown here, we notice that release of oxygen and binding of carbon dioxide on the right is favored by an increase in H+ concentration, that is, by a drop in pH. This drop in pH favors the protonation of key amino acid side chains that result in release of oxygen and binding of CO2. This occurs in tissues and is favorable for hemoglobin function in that environment, delivery of oxygen to the tissues requiring it, and pickup of the metabolic waste product, carbon dioxide. Another way of looking at this is that the acid-base properties of hemoglobin are altered as a result of the conformational changes associated with oxygen binding. Look again at the equilibrium equation for the binding and release of oxygen. Looking at the left of the equilibrium, we see that oxygen binding would be favored by release of protons, that is, by a high pH environment. The oxygenated form of hemoglobin is a stronger acid. Certain amino acid residues exhibit a lower pKa value and are therefore more likely to release protons. From our table, we see that this occurs in the lungs, where the pH is higher than that of actively metabolizing tissues. So then, the pH environment in the lungs favors oxygen binding. In actively metabolizing muscle, there is a drop in pH as a result of the generation of acidic waste products. As the concentration of H+ increases, the equilibrium is driven toward the right, toward release of oxygen to the tissues and binding of CO2 waste. Perfect. If we compare the oxygen saturation curves for myoglobin and hemoglobin at varying pH values, we see that hemoglobin's curve shifts further right with each drop in pH. Remember, this means that the 50% saturation of hemoglobin is getting larger as pH drops, meaning reduced affinity. Notice the curve for myoglobin. The same curve results at any of the pH values examined. In other words, it remains unaffected by these mild changes in pH. Another factor that influences the binding affinity of hemoglobin is the presence of an effector 2,3-bisphosphoglycerate, or BPG. As you can see from the structure of BPG shown here, it carries multiple negative charges. It's not surprising, therefore, that its binding to hemoglobin is electrostatic. On the right, you can see a representation of the crystal structure of hemoglobin bound to BPG. Notice the negative charges on BPG and the multiple positive charges from amino acid side chains extending into the binding pocket. Let's next look at any changes to the binding curve in the presence and absence of BPG. Notice that in the presence of BPG, the blue curve, the curve becomes more sigmoidal and has shifted right toward reduced affinity, as compared to the affinity in the absence of BPG in the red. This might seem contrary to purpose, but remember, hemoglobin needs to be able to release oxygen more readily in tissues, and this feature allows it to do so. Another interesting feature of hemoglobin is that it has a different fetal form, known as HbF, as compared to an adult HbA. If we compare the binding curves of HbF and HbA, we see that the fetal form exhibits a higher affinity. The maternal hemoglobin will encounter no difficulty in binding oxygen in the high-pressure environment of the lungs. The important consideration here is that oxygen is delivered to the fetus. Notice how perfectly the difference in structure suits this purpose. In tissues, the maternal hemoglobin has reduced affinity and the fetal form has higher affinity, thus ensuring delivery of oxygen from mother to fetus. What a sophisticated system for oxygen delivery, yet certainly necessary for organisms whose viability is absolutely dependent on oxygen.